Rosalyn Moran, who has moved back to London and is at King's College London. Rosalyn has done fantastically intriguing work using models and pharmacological manipulations and other clever experimental manipulations to look at parameters and the emergence of aberrant dynamics and behaviour. So Rosalyn will be speaking on models of behaviour and how they can be integrated with brain connectivity for a computational psychiatry. Thank you, Tanya, for swapping over to Rosalyn, and thank you, Rosalyn.
Thanks, Michael. Hopefully you can see my slides now. And I'd thank, first of all, Randy and Tanya for getting up so early or either not going to bed at all, possibly. And also to Michael for staying up late and chairing this session with this great beard that's giving me a very good Sigmund Freud sort of vibe. And yeah, so today what I'd like to discuss in 3.5 slides and a few minutes is whether we can bring some of our brain connectivity methods to clinical relevance in psychiatry soon. And I guess the context is the global context of vaccines for COVID that we've been able to produce in record time. So can we put the same urgency behind the imaging and modeling methods that we're discussing here into addressing easy clinical questions, or maybe so-called easy clinical questions, or more deep clinical questions on the causes of psychiatric disorders. So what I'm going to do is frame in a very sort of thin overview and review the connectivity evidence and theory of in schizophrenia. And so what I wanted to frame it on was predictive coding. So this brain connectivity in schizophrenia, if we think about it, from a theoretical point of view, and Carl Friston has updated after 20 years his disconnection hypothesis with Chris Frith in this wonderful paper in 2016 in a sanguine view, as is his way, describing how the systems level neuroscience and also the molecular and genetic neuroscience are converging into an explanation for schizophrenia that really focuses on synaptic efficacy, and specifically synaptic efficacy mediated through the NMDA receptor. But starting from the systems level, what you can see if you think about the theory of cortical connectivity is emergence of symptoms in schizophrenia, and also things that patients with schizophrenia do better than healthy controls. And so this predictive coding account is a process model for the normative model of free energy minimization, which is sort of inspired by the Helmholtz idea of having an inference engine between our ears. And so the first part of the disconnection hypothesis really starts with synaptic efficacy changes in low levels of the cortical hierarchy around sensory regions. And so the idea is that under predictive coding, we have predictions traveling top down from higher to lower cortical regions, sending predictions of causes of things in the environment. For example, there's a dog that's filling our visual scene. And so these predictions will then be trying to explain away prediction errors until we perceive that dog. And if we have changes in our perceptions, then our predictions will be slightly off. And so we'll have to send a signal up the hierarchy through a prediction error, which can then be mediated by new, better predictions to then shut down this circuit. And this backward prediction for a prediction error might be mediated through a canonical microcircuitry, incorporating different sorts of pyramidal cells, deep for the feedback, superficial for the feed forward, and then crucially, the volume of the different prediction errors. So how much you listen or pay attention to each of your sensory channels, or indeed parts of the hierarchy, is crucially controlled by a second order mechanism, and may indeed be linked to the solenoidal flows you've just been discussing, but certainly linked to features of neuromodulation, particularly at superficial pyramidal cells. And the idea is that they are either high or broken in the early sensory regions of schizophrenia. So therefore, what you have is your radio set onto all the channels at once. So you're sort of jumping through different AM stations And you have this aberrant precision or abnormal gain control that's giving you very confusing sensory prediction errors. What happens as a result then might transmit to the frontal cortices. So you have to make predictions without being able to attenuate sensory errors. What you then might do is compensate in frontal regions and so have aberrant or hyper precision in deeper levels of the hierarchy. And this might then contribute to interesting things like altered agency, so ascribing things that you did to external people because you're not able to downweight your sensory prediction errors. And so this scheme of forward and backward message passing is crucially dysregulated in schizophrenia. And the key culprit is thought to be not dopamine, but NMDA, the NMDA receptor. And there's lots of genetic support around this. So looking at mutations and variants in GWAS studies that Carl has reviewed in this paper, what you see is that there's lots of modulations of the NMDA receptors, so genes that are known to be involved in this pathway. And also, interestingly, things like CACNA1C, which are voltage-gated calcium channels also interacting with NMDA. The paper makes the claim that the structural connectome changes are secondary to this synaptic efficacy change, so that you see changes in activity-dependent myelination potentially, and also pruning because of the changes in receptor levels and their functional efficacy. And so secondary changes in white matter tract might occur later. And also that you get this sort of idea of EI balance in schizophrenia, again, as a more secondary effect that might be showing some of the NMDA effects, particularly at GABAergic interneurons. And so just to give you a little example of how we looked at this circuit before, we didn't get a full view. We were looking at a rodent model of oscillations and how they're perturbed by ketamine. So ketamine, of course, is an NMDA antagonist and a psychotomimetic, and its discovery as a psychotomimetic is what led people to the NMDA hypothesis itself. And so what we looked at in this study was the spectral effects in local field potentials in embedded electrodes in animals, and show that you do get this increased gamma and this decreased theta oscillation under enhanced levels of ketamine, and that this did cause aberrant changes in precision and a subsequent decrease in top-down connectivity from the medial prefrontal cortex to dorsal CA1 in the rodent hippocampus. But interestingly, that a lot of the changes in synaptic efficacy that we saw in these data were actually in the hippocampus rather than the medial prefrontal cortex, and mediated by our models of GABAergic interneurons. This is through a dynamic causal model. But even though this paper was incredibly sanguine in putting it all together, looking at the more reviews from more specialized literatures on, say, oscillations or functional connectivity in schizophrenia, it seems that the picture is a little more fragmented, though I think hopefully one would go with the sanguine view. And even Peter Oldhouse, who's a long history in studying gamma oscillations in patients with schizophrenia, has recently sort of cautioned on findings in gamma, showing that actually there may not be this sort of trajectory of gamma associated with first episode psychosis and chronic disorders that he first proposed, and that ketamine and schizophrenia have pretty different gamma qualities when it comes to their hyperactivity. So this is obviously not a perfect model, and indeed is probably all, so these features of oscillations are just secondary to the synaptic changes. So if you look at schizophrenia in functional brain connectivity through fMRI, there was a lovely review in the same year by Dana Bartsch, and what she found was by studying actually the cognitive symptoms, so these are sometimes less well studied as opposed to the positive symptoms of psychosis, but looking at the cognitive abnormalities in schizophrenia, generally what she found was that there wasn't any unique relationship between cognitive symptoms and functional connectivity, though generally there was a predominance of frontal disconnectivity, though she does conclude that there's no clear hyper or hypo connectivity except possibly with frontal and subcortical regions. But what she did is put a nice sort of schematic together to try and summarize the literature that she reviewed, describing how task positive and task negative regions, so task negative here in red, the MPFC and the parietal cortex, task positive in the dorsal ACC and the DLPFC, that these tend to have different connections than you see in healthy controls. And also that if you were to extend this circuit to other solid literature that's linked to cognitive deficits, but in no unique way, it would also include thalamocortical and cerebellar thalamocortical loops. And also positing again in the context of lots of reinforcement learning tasks that the striatal circuitry and its link to thalamus and prefrontal cortex is another key aberrancy when you look at functional connectivity in patients with schizophrenia. And so I'm going to briefly describe just one experiment that we've been looking at recently with Suki Shergill at King's, looking at patients who were resistant to dopaminergic treatment. So these are refractory patients where there's there again, its own literature, which is interesting, considering whether these patients are a subtype or an extreme of schizophrenia. And what they'd shown before in mass univariate analysis of fMRI data was that treatment-resistant patients had impaired learning in reward learning tasks, but actually have sort of retained some of the striatal activity that you see in healthy controls. And so what we did was apply a dynamic causal model to the sensory learning interaction and the control in the regions involved with this particular paradigm, which was an emotional two alternative first choice tasks where there were emotional faces and neutral faces, and patients had to select one for more predominant reward. And what we were looking at was the cingulate cortex and regions of the striatum and thalamus, as well as the amygdala and fusiform face area, which were involved in processing the faces. And we found a normative network that had very strong connections, particularly from the ACC to all the subcortical and cortical regions, and the sort of also reciprocal feed-forward connectivity from these regions into the ACC. And interestingly, when we looked at the treatment responsive patients, what we found was consistent with the literature, this sort of enhanced drop off of the ACC. So the ACC doesn't really stay in the game when you're looking at these faces and making rewards. And it also doesn't connect as well to the caudate as compared to healthy controls, but you do seem to have intact cingulate connectivity to the amygdala and the fusiform region, whereas the treatment-resistant patients actually showed none of the subcortical changes or indeed cortical changes, but just had a much greater impoverishment in the rumblings or perhaps even turbulence of the anterior cingulate cortex. So we'll try and come back to that in a second, but just to bring you on another whistle stop of electrophysiology and how else we could look at brain connectivity, if not in the context of complex cognitive tasks, but in the context of a very simple visual grating. So here, this is a work from Alexander Shaw, who looked at MEG data from patients and found that there was decreased frequency and decreased power in V1 when showing gratings to these patients. And we worked together to build this DCM, which looks at different cell types, and found actually that it was the neuromodulation through interneuron self-connectivity in the superficial layers that most explained this pattern of gamma changes. And then just to link it back to try and justify using electrophysiology, or to try and think about comparing what you get from MEG in this case to fMRI, this is an example of a mismatch negativity paradigm not applied to schizophrenia patients, but applied to genetic channelopathies, patients with changes in potassium and calcium channels. And it's a CACNA1 family, but not the same family that is implicated in schizophrenia, though it sounds the same. It's a slightly different voltage gated calcium channel. And what we found, if we did a DCM, again, having these sorts of detailed cell models from electrophysiology, that we could somehow identify changes in channels based just on the properties of a simple ERP, or event-related field, in this case of MEG data. So just to open the discussion, the question is what we can do with these selection of connectivity methods from DCM and resting state to, do we just look at the ERP and channel CZ and leave it at that? or do we go in and build in these DTI metrics, look at specific neural mass models, and so on and so forth. And so just for you to ponder, the questions are: what clinical applications could be addressed soon? Are connectivity measures assessing symptoms or causes? Why is a simple frontal connection so difficult to reproduce? And if it was reproduced in the context of schizophrenia, would it be useful? Then things like, do tasks exacerbate problems of fragmented literature or potentially provide better symptom deconstruction? You know, is unraveling this disconnection hypothesis a pipe dream given all its beautiful compensatory and extra secondary follow-ups from this core NMDA deficiency? And then, you know, maybe EEG is where we should be putting our money, since also it's cheaper and potentially it's closer to genetics. So with that, I will stop sharing. And thank you all. Sorry, I had a thank you slide, which is all your names and my participants.
Wow, thank you, Rosalyn. You've thrown a lot of questions out there. Bamboozled as the chair.
Sorry,
My thoughts disoriented, but thank you. A beautiful overview of you know, both the unifying framework of connectivity in schizophrenia, and then unraveling it with all the various pieces and components of gain control and synaptic plasticity. While others gather their thoughts, I mean, one question that is going to come up as well, and one thing as a clinician that, you know, we need to find little hills at the base of the mountains that we need to climb to bring computational psychiatry into the clinic. And as you know, schizophrenia, and as you showed, has a very heterogeneous profile. And yet we always sort of start with the same antipsychotics, and then we always sort of like work through these algorithms. And patients presenting very differently, some with profound negative symptoms, some with apathy, some with reality distortion, you know, very fixed paranoid delusions. What's your thought on a computational assay that might partition patients into different pharmacological deficit states, and therefore we could at least be better than current practice at choosing the first or second antipsychotic? Is that a reasonable proximal goal, or what are your thoughts on that?
Yeah. So I think, although because I have moved to King's, PET is more of a thing, and even though it's massively expensive, so there's now the first in clinic levodopa tracer to look at striatal occupancy. So it may not be a computational psychiatry solution, but is an imaging solution that might look at striatal dopamine and at least look at whether they're likely to respond to first-line antipsychotics or not. So in these treatment-resistant patients, we may be able to identify that the dopaminergic D2-centered antagonists are not appropriate. But that's not using all our wonderful repertoire. So I think another question of staging, which I guess I was more enthusiastic about before reading Peter Ullhaus's review, was the gamma story, so that the gamma oscillations and where efficacy had changed from changes in gamma in V1 to maybe more profound changes in the prefrontal cortex, that might help identify, you know, if this is a sort of, along with your symptoms, someone who's initially having these sensory disturbances, or if they've gone into the full compensatory mechanism where they've just rearranged their neurobiology to try and cope with all these volumes turned up. So I thought gamma might be the way to go there, but now that looks a little, although, you know, like you said, this literature is very difficult because if you have a heterogeneity of findings, there's always the possibility that it was just the heterogeneity of subjects in the first place. So it may not be the method itself, but rather what's underlying the biology. But what I think is a promising way for trying to get to the next level of therapeutics, so beyond the drugs that we have available now, might be to focus more on electrophysiological measures of synaptic efficacy. I think in some of the tasks that people know to have changes in schizophrenia, like the mismatch negativity. And then also there's interesting work coming out on sleep and whether we can characterize spindles and use those as an endophenotype, which will definitely take a lot of modeling to try and look at whether there's particular subgroups of our patients that have particular channel deficits. And sleep seems like a kind of clean electrophysiological readout that might help in that regard. So I think what I'm trying to say is, I think for the simple question of whether people, well, not simple at all, but for some markers of responsivity to first-line antipsychotics, I think there's already things you can use, though PET is expensive. So I think what the computational psychiatrists like myself might do, who aren't actual psychiatrists, as you know, will go and try and build methods that get more to the cause rather than to the second level features that may in fact be dopaminergic.
Okay, well, thank you. And then I guess related to this is a question that's come up in the question and answer box. I think it relates nicely to this, that we're still basically targeting psychotic symptoms with anti-dopaminergic drugs. And we haven't realized the potential of the NMDA pathway for new therapeutics. Is this because we're trying to treat the reward system, up or down regulate that, just to make, you know, the brain work in the face of this pervasive NMDA channelopathy? Or what are your insights on this question?
Yeah, I think because we haven't found the right agonist for an NMDA to try and reproduce the features, and probably because and this is an interesting challenge, is that we're unlikely to treat schizophrenia prophylactically. So, you know, even though we might be suspicious that in a developmental trajectory that there are signs of first episode disturbances, you know, are you going to go in and try and alter the NMDA receptor? At the moment, we don't have very good ways to do that from the pharmaceuticals. And I think the dopamine it's interesting that you're thinking regulating just reward and then, you know, people might behave better. That's a way I don't think about it. I think it's the sort of psychotic symptoms are helped and alleviated, but rarely any of the cognitive symptoms are affected. And it's probably because we're treating the disease too late, but also it's difficult to think how we would treat it early enough without having really, really reliable genetic or biophysical markers that this person is definitely going to go from clinically at risk to full-blown psychosis. I mean, I'd be interested in your own clinical point of view on how that might go down with the parents of a 17-year-old who, even if we did have a really good NMDA receptor drug. But so I think even though we don't have a lot of animal physiologists in this meeting, that their work is crucial to try and understand longitudinal changes over development, and particularly in the late adolescent period. So we need better animal models as well. And they need to come to this conference because they don't often think about it in this way.
It's a big discussion here in Australia, actually, because there are a lot of clinics that are following high risk and not even at-risk mental state. And what are these brief psychotic symptoms? There's a little word for them. Yeah, and then the antipsychotics that we've got, you know, they're metabolically harmful. So obviously there's different schools of thought as to the risk and harm and benefits of doing this. But I'll turn it over to Dan Lurie, who's put a question, and then we might go to some of the panelists. Dan, do you want to ask your question off mute?
Sure. Thank you, Rosalyn, for this really kind of overwhelming tour of what we think might be going on in the brain in schizophrenia. And I really like the question you opened with in terms of contextualizing what humanity's been able to do so quickly to come up with a COVID vaccine and what we might be able to accomplish if we were to pour the same sort of resources into like a translational and computational psychiatry. And I'm curious if you or the other panelists have thoughts on the degree on that analogy in terms of, how well we currently understand the immune system and how well we currently understand the brain as a system, and whether the brain can kind of be understood mechanistically in the same way as the immune system does. I mean, it's hindsight is 2020. Well, now that we kind of know how the immune system works a little bit more, it kind of makes sense. And maybe at some point we'll get to a point with the brain where we say, oh, well, of course it works that way. And it's simple once we know the principles. But it also feels like to some extent, a lot of the work that has been talked about this week is talking about how the brain is really this complex dynamical system that maybe the immune system is in some ways, but that we can intervene a little more easily in the immune system, and whether there's kind of a qualitative difference there in terms of the types of systems that we're working with.
Wow, great question. And I mean, is Carl still here? Oh yeah, so he's been writing some stuff on immunology recently. But what I might say is, I think, from a broader perspective of what the governments around the world did was, put a lot of money in parallel so that someone would win the race, and there might be a vaccine even, happily, lots of the investments paid off, and we have more than one, though they're unequally distributed. But so I think, I would take that figure at the end of Carl's review, look at, there's not thousands of genes that have been implicated in the GWAS studies. We do know it's, they're all going to have small little effects, but I think we can go a little bit farther to understanding how many or what sort of combination gives you this tipping point where now your synaptic efficacy is just blown. And you could have, even if you had a small change in your calcium dynamics, for example, when you were 16, that would have settled the system, allowed you to learn and not go into this state. So maybe it's being naive to say that I don't think the brain is so different to the, now I know we better understood beta coronaviruses probably than what voltage gated calcium channels do in neural circuits. But I would say that, yeah, we just have to do, but we kind of all have to converge on the same question, which is what if you also think of everyone wanted the virus to keep the spike protein out of our lungs. So what do we want to do in schizophrenia? And I think that's maybe where the field has to kind of move in one direction. And because, it's not for sure, if it's for sure not the case that lots of people giving talks on schizophrenia would all focus on the synaptic efficacy story. Lots of people will have focuses in different frameworks. And so I think bringing a consistent framework and a consistent question is something that the vaccine challenge didn't have to handle. But I do think if we had that consistent framework, that I do think you could, and again, with rodent models, just knock out some of these genes and see 4 of these lead to this, 6 of them, you get this massive effect, and have the understanding of what they do in schizophrenia. Treating them is also hard, but I think if we had that specificity, the pharma companies, like you saw in the vaccine race, would come behind us.
Okay, thank you, Rosalyn. Yeah, I share this very interesting analogy, and I was just reflecting recently on my youth when I read Gannon's Physiology, and then reflected how remarkable it was in the 70s and 80s that many of the organ systems in the body, the basic principles, and even down to the different cells involved in the different loops, particularly renal physiology and the homeostasis of blood pressure control, were really worked out. And, it was a really systems approach, all based on homeostasis. And yeah, we just need to do that for the brain, but maybe we're making progress. I'm going to go across to one of the panelists. Randy has had his hand up for some time. So do you want to unmute, please, Randy?
Thank you, Michael. A lot of points that you raised, Rosalyn, which I quite liked, and I don't want to go through all of them because it's probably more than we can do in 25 minutes we have left. One of the things that came up yesterday, and I think you were gone by then because it was getting late, but there's a paper I put in the chat window that has to do with white matter and the emergence of psychotic episodes in development. And it kind of connects with the comments you made about the work that Carl had put together, where you get structural changes after the functional changes. So the emergence, I think if I remember correctly, that the emergence of symptoms you were kind of suggesting doesn't actually depend on structure, it depends more on the function. So I was thinking about this in the context of building, use the word that Victor likes, manifolds, or I like as well, or a landscape that shows sort of the capacity of the system to go in different sort of configurations or functional configurations, and whether you can sort of envision, because this is the emergence of psychotic episodes in schizophrenia is often a developmental trajectory as well. So there's sort of an event that happens, whether it be biological or environmental, that the person then transitions to a deficit of some sort. And whether you can envision sort of using the combined methods you're talking about to sort of, especially for people at risk, to see if you can track out sort of the emergence of this pathological manifold that will eventually then transition to a psychotic episode.
Yeah, because I think that's the right approach, is to look at across the platforms in the manifold space. Like, and I am thinking of TVB is where. So even though it was, that the white matter tracks is it's an effect, not a cause. So the cause is a synaptic efficacy. But nevertheless, there is like a lot of consistent literature, and Carl in that paper cites Ed Bulmore's work, which has, 2 major white matter tracks, and it's, the places that you would think. So at the frontothalamic cingulate, and connecting insula temporal cortices and occipitotemporal cortices. So I think putting those focusing there in your manifold-based description, and again using the sort of methods Brandon and yesterday's theme of compression. So trying to look at those features in a very abstract way, using all the good neurobiology of the structure that's cause and effect. It's sort of it's important when you're looking at a trajectory. So is it coming on? When is this now starting to happen? Versus the maybe functional connections that might look aberrant sooner. So I think that the anatomy is very important to do that initial sort of dimensionality reduction. And I know you can't have like half a TVB, but let's imagine you could strip out some scaffolds and be like, just focus there. And then in terms of the longitudinal analysis, I mean, this is what absolutely needs to be done in these multimodal assessments. They're just wildly expensive. And we don't have great predictors of who's going to develop schizophrenia. They're pretty good. But, I think Klaas, our friend in Zurich, is doing a good job recruiting people. And I know King's has huge databases where, they're doing this in simple cognitive tasks, but it's just too expensive to take 20,000 people from the UK and do it longitudinally. But I think I wouldn't throw away any modality at this point.
Yeah.
I know that wasn't your question, but
could you envision perhaps, I mean, I know that you're a fan of EEG, for instance. So could you envision, for example, using a low res version of that? So if we do sort of like the full Monty version of capturing all these data and putting them into something like TDB or an equivalent, and then deriving from that sort of a low res simulation that would tell us what some of the EEG signals would be for a person at risk, then be able to use that much the same way as you use like a blood pressure cuff to monitor transitions that may be approaching using something like a low res EEG.
Love that idea. And then so you have your sort of good full model on a server somewhere, and then all you're taking to the community, if you like, is a few electrodes and a cheap system. Yeah,
I mean, if we're gonna transition to something that's usable in the community, we're not going to be able to use an MRI for that, obviously. So that's why I say EEG.
But there's a machine called a hyperfine that looks like, have you heard of this? So this is a ultra low resolution 65 millitesla scan that's doing all kinds of weird and wonderful things, just structural images with some deep learning associated with it. That's £60,000. So this is that I'm working with the Gates Foundation on a totally different related thing related to schizophrenia, but so there might be some structural MR features that would get into the community through this sort of technology. But again, they'd be literally like low field, but actually have some pretty decent properties. And then with the combined EEG, I mean, I think we have to do this, and that if we had the right power, you'd see pretty clear signals. Yeah. I think that's what all of these reviews say, is that a lot of these studies are just a little bit underpowered.
Cool. Thanks, Rosalyn.
Okay, Andre Lupi, do you have would you like to unmute and ask your question?
Hi, sure, thank you. This was really intriguing work, and in the vein of what you have been telling us about, I'm wondering if you have any particular thoughts and what they are on using models directly to characterize and stratify patients. So on one hand, one can have computational models, so in the vein of computational psychiatry, and maybe a large battery of them where there is 1 or 2 parameters that can be fitted for each patient to identify some kind of phenotype there, but also then computational generative models of the kinds that we have been hearing about throughout this workshop, so that we can have both behavior model derived parameters and brain model derived parameters to understand the space of these subjects.
Yeah, I think that is a focus of computational psychiatry. The sort of panacea is that you don't even need a cheap MRI scanner or a cheap EEG scan. You can just do a sort of behavioral task on your iPhone, and if you get the right sequence, then you know you're accessing dorsolateral prefrontal cortex because it's been validated before. So using tasks to literally map features of neurobiology is another big push around computational psychiatry. So, you know, people like Peter Dayan would work on depression and, you know, features of negative reward learning and uncertainty and so forth to try and get metrics of maybe serotonin or noradrenaline. I think that work is going on in parallel, and I guess it depends whether, well, what level you want to understand the neurobiology, whether you have to scan in parallel with these behavioral batteries, or whether you can just look at the behavioral battery and then say the disease course and say, you know, people are recovering from depression because you see their, you know, language is more fluid or more diverse or so forth. So I think the, so Dan has, so the computational psychiatry and movement is hoping that at some point we'll have 6 tasks that will assess psychosis, 6 tasks that will assess depression, you know, another 4 tasks that will try and look at prognostics in autism. But it's very, very early, because I think we probably need richer tasks than we have already. You know, we don't have the turbulent manifolds of Gustavo Deco embedded in any simple press button response time yet, but I think Brandon's work yesterday helps to link those manifolds, and it might help us move out of the scanner and into patient classification on a wider scale. Yeah, behavior is crucially important, and taking it very seriously. And in schizophrenia, and in this review by Carl, and we've been doing a lot of it recently, is eye movements is another sort of cheap and easy way to try and look at potentially like working memory, prefrontal dopamine, you know, striatal dopamine, while just looking at where their eyes go. But so really specific models of behavior, which I haven't put in the talk, are another component.
Okay, thank you. Yes, because of course the cognitive symptoms of schizophrenia are the really socially disabling ones, not necessarily the symptoms that we see in the clinic. Vic Yersa, has a question. Would you like to unmute yourself, please, Vic?
I have done so. Yeah. Hey, Rosalyn.
Hi, how are you?
Nice to see you. Good.
Nice to see
good.
you.
Aging. Wonderful talk. Thank you. And it's actually right on the spot with questions that are very hotly being discussed in the European community, you know, your neighbors. Yeah. And if you look at the upcoming programs for funding of the next 7-8 years in the EC, you will find that mental health and the treatment of mental diseases is really the hotspot, yeah, together with aging. And in particular, their questions are that are being raised are we're talking about many hundreds of millions of euros, are about curative approaches towards mental diseases. And I was wondering, in the context of what you have presented, in the context of your work, you're using DCM, which is a model-based approach, but mostly of diagnostic, with the diagnostic objectives. You want to figure out what are the cause-effects relationships. And you also addressed this earlier in a question that you're looking at the causes of this. So I wondered whether it is with this type of approach, now that you have a model-based window towards the disease, can you also explore not only diagnostic, but also alternative, maybe out-of-the-box curative approaches that would not be targeting directly just the removal of the cause? What I have in mind is what we are doing in epilepsy and TBB, as you know. There we are also trying to identify the epileptogenic zone. The curative approach is you remove the epileptogenic zone. But having a causal model in the hand, you can also look at you can explore other possibilities, such as stimulation. How can you stimulate different areas, not necessarily just the epileptogenic zone, in order to stop the seizure from propagation? And through this, you can actually stop the expression of the clinical symptoms, which would actually help the patient, not cure the disease, but provide potential semi-curative approaches. You can do this also with micro lesions, yeah, not just a full resection of the epileptogenic zone, but remove or take a disconnective approach with the knowledge of where to target actually the disconnection. Is something like this possible for schizophrenia or other potential disorders? With the approaches that you take, can we envision the targeting of novel curative approaches that are not just having the objective to remove the cause of the disease?
Yeah. I think there's easier places to start than schizophrenia. Possibly, you know, it's just coming to mind your generative model, TVB framework, the connectome ideas of, you know, the Helen Mayberg work in refractory depression might be, you know, where that approach is, you know, you're not doing anything to serotonergic systems or learning systems. You know, you're stimulating part of these pathways from prefrontal into the ventral striatum, and, you know, you're getting whopping effects in some people and not in others. So, I mean, I really like that approach. And again, it's hard to describe, well, it's not hard to describe, but this is the community where people understand the redundancy and non-linearities, where you can change something not to, you know, put back in or take out the pathology, but just to basically use a redundant space that you were going to perform that function with using something else. And so I think that's not well developed in, I know there's a little bit of literature using TMS in sleep in patients with schizophrenia to try and improve memory consolidation. This is linked to the idea of, you know, spindles are a decent endophenotype. They seem to have some of the genetic markers of schizophrenia, but that's only kind of 1 or 2 papers.
It doesn't have to be stimulation, Rosalyn.
Yeah. No,
Just an
sorry.
example. Yeah.
No, yeah, I know. Because I mean, you know, if we can never get the right NMDA drug, then like target something else that can also do synaptic efficacy, like calcium drugs. So, or some of the serine pathways, or so. Yeah, I think it's a way to view the problem, so that you have multiple paths to curing that aren't just the, you know, replacing what wasn't there, or, you know, dampening down what's too much, but putting in a something else, be it pharmacology or, you know, a behavioral intervention. Like maybe there's, let's talk about the schizophrenia and say the treatment resistant versus non-treatment resistant. You know, I think it's interesting that the treatment resistant don't respond to dopaminergic meds. So, and in my view, I've just sort of constructed this model where people get so sick that they don't need that sort of hyper-dopaminergic tone to change the brain. So they get schizophrenia even without, you know, dopamine doing anything wrong. But let's say, how did we, so if schizophrenia is developing into late adolescence, then, you know, maybe we just make sure people don't, you know, do very adventurous things, so their dopaminergic system isn't challenged, or they have a nice time at school, and, you know, those kind of interventions that you might change their neuromodulatory reactions in a sort of learned way by a safe and protected environment. You know, this is kind of fanciful, but this is a way that you may not have the final hit of dopamine that brings you into psychosis. And I think that's what clinicians probably know, but putting it in a formal framework as a cure, you know, haven't seen that done. But I think that's the only way we can probably really hope to cure diseases is by taking secondary routes to the problem. Yeah.
Thanks, Rosalyn. Our next question comes from Carl Friston. If you'd like to unmute yourself, Carl.
Yes, thank you very much. I was just going to sit back and admire this one, but I can't resist so endorsing Rosalyn's answer and taking up some of the excellent challenges in Victor's question, just practically. And one thing that came out of Rosalyn's presentation that was picked up by a single word that Michael used was the notion of a channelopathy. And I think that's a big shift. You know, if in the 21st century, and in fact in the third decade of the 21st century, we now conceive of schizophrenia as a synaptopathy, and in particular developmental synaptopathy, that is a big shift. And it sort of begs the question, well, okay, if you understood the synaptopathy that underwrote schizophrenia to the same level that we understand Huntington's disease and Parkinson's disease and certain genetically acquired epilepsies, how would you treat them? And of course, it's very, very difficult, even if you really understand the nature of it, to, you know, to treat these diseases. And I think we're faced with the same problem with schizophrenia. So, you know, I don't know that sort of even micropsychosurgery is going to work. Drugs by themselves are unlikely to work in the sense that, you know, all of these things have been tried, stem cell implants in Parkinson's disease over the past decades. None of these things have worked. So what has worked, or what could you imagine working? And I think that the epilepsy model as the poster child of a synaptopathy is probably the way to think about these things. So, you know, Victor, just in response to your question and strategic, you know, considerations from the point of view of the HBP and the, you know, the next funding round, certainly as you know, in our conversations, we've elected, well, I have elected anyway, to sort of promote the synaptopathies that underwrite certain forms of epilepsy as a proof of principle, in the fond hope that that could then be translated into something which is more complicated, like schizophrenia. So what could you do if you had a perfect mechanistic modeling understanding and of epilepsy? You can do a lot more than with schizophrenia, because you can do sort of neurosurgery and sort of, you know, if you can identify the you know, the epileptogenic zones. But there's another thing you can do, which is what Rosalyn was talking about, which is, you know, you could try to predict those points in time when you get this wandering in parameter space on parametric manifolds that contain the synaptopathy, and start to warn the patient when they're likely to have a seizure in the next few days or have a psychotic break. You know, so, and at a longer time scale, I think you can tell patients and their parents what might happen to them, you know, in terms of long-term trajectories. So, I'm not sure there's going to be a cure, but there are certainly lots of really useful moves you can make if you can get a strong empirical handle on the trajectories of the synaptopathy at different points, both neurodevelopmentally and in terms of responding to life events, and understanding things like resignation syndrome and the catatonia that you get with that, which is very similar to some old kinds of schizophrenia. So, and another strategic note, Victor, I was just thinking, sort of Katrin's notion about language being an important phenotypic expression of things that may underwrite mental health. That's an underexplored, cheap and cheerful bedside way of getting at sort of the message passing implied by the kinds of synaptopathies that you see in schizophrenia that could accompany eye tracking, smooth pursuit, mismatch negativity, pupillary dilatation, in addition to all the standard bedside earn beads tasks and all of that. So I'm saying this because practically, Victor, it might be a good idea to loop Rosalyn into the discussions with that you're having with the HBP in terms of sort of. But also just to really endorse this notion of channelopathy and synaptopathy as the common convergent point. I'm sorry being indulgent, but I was intrigued by the question about the immunological perspective here, and certainly in the past few years, the differential diagnosis of somebody comes along with a sort of an autoantibody presentation, a sort of anti-NMDA receptor in A&E includes schizophrenia. So there's an increasing awareness now of all the convergent pathways on the particular kinds of synaptopathy that cause schizophrenic symptoms. That would certainly include the immune system. There are likely to be other ones as well. Again, speaking to the importance of thinking about this as a synaptopathy, but can be broken in many, many different ways. In the same way that Parkinson's disease, you can start off with Lewy body diseases, and it's understanding the multiple routes to this may be important in terms of prognosis and diagnosis. Is that what you meant, Rosalyn, by sort of having a safe environment and just understanding what is likely to happen to this person in the next few months?
I think the epilepsy analogy is great. So, if someone's tired and overworking and they have an epilepsy disorder, they might be more likely to have a seizure and, back off a little. It's the sort of same idea that you have an environmental intervention that's because we're aware of what might happen or where the tipping points are in the load of the channelopathy or the synaptopathy. Yes, exactly. And trying to reverse it is harder, but trying to counteract it by slowing down literally is sort of what I meant.
Yeah, prevention, not cure, personalized medicine,
yes.
And all that good e-health that sort of attends it, the Fitbits you can wear. So you can now have an fMRI, a wearable fMRI now from Bill Gates. That's good, isn't it?
Yeah, it's about the size of a desk, and you just stick your head in it, and you can get a pretty good signal. Yeah. They're the new black.
What will they think of next?
Yeah, yeah.
Yeah, but as well, of course, with wearable watches, pulse oximetry, possibly. We're all looking at a camera here, pupillometry, skin conductance, which is a little bit more invasive. These are all the sorts of simple sampling technologies that we could harness to help people track their own, the ebbs and flows of their own psychotic disorders, which usually do change quite dynamically. And blips was the word I was looking before, the brief, limited, intermittent psychotic symptoms. And that's a great example of somewhere where perhaps you could, through external feedback, help people improve their homeostasis in the face of are not yet fully entrenched channelopathy.
Yeah.
Any closing remarks, Rosalyn?
No, yeah, we want to skirt around the channelopathy and then hopefully fix it while we're managing. Let's do this in 10 years though, not like 50, because that's too long. That's my closing remark. Thank you for your attention and for the really brilliant discussion. Enjoyed it.
Okay, well, look, thank you very much, Rosalyn. A beautiful talk and a intriguing discussion.